you ever dreamed of diving into a living, colorful world under the sea? Of witness a silent, swirling city of life? For millions, a coral reef is a journey of a lifetime. But this breathtaking beauty is fragile, and it's vanishing before our eyes. Let's start with a question. Why are corals so crucial to us? The answer is all of the above. Coral reefs are the unsung superheroes of our planet, but every superhero has a weakness. Rising ocean temperature are their greatest threat. Since the 1950s, half of the world's live coral has vanished. If temperatures continue to rise, we could lose most of our reefs by 2050. But what if we could give our hero a shield? Scientists from the Chinese University of Hong Kong ask the same question. And their solution is as ingenious as it is unexpected. You've probably heard of probiotics, the good bacteria that keep our gut healthy. The corals have a microbiome too, a whole universe of microorganisms living on and inside them. And just like us, their health depends on these tiny partners. So, the big idea was, can we give corals a specially selected good bacterium to help them survive heat waves? The challenge? Most probiotic bacteria we give to corals are like temporary visitors. They help for a bit, but then they leave. For a lasting solution, we needed a long-term resident. So, instead of looking for the strongest or fastest-growing bacteria, they looked for one that wants to stay. And by reading the bacteria's DNA, they found a perfect candidate, Ruegeria MC10. It shows two key traits that make it the ideal partner. Trait 1. The unstable, self-rewriting blueprint. The discovery of an explosion of jumping genes known as insertion sequences. Think of them as genetic copy-paste errors that run wild. These jumping genes randomly land in new spots throughout Ruegeria MC10's DNA. When they jump into the middle of an important gene, they break it, turning a useful instruction into nonsense. But the impact is even bigger. Because there are so many identical copies of these jumping genes, they can cause large-scale rearrangements, deleting, flipping, or shuffling whole sections of the genetic blueprint. This creates a highly unstable genome, supercharging its ability to evolve rapidly. It's a powerful, though risky strategy to quickly adapt to a new lifestyle, in this case, life inside the coral host. The bacterium is actively and radically rewriting its own blueprint to become a permanent partner. Trait 2. The incomplete toolkit. To live independently, a bacterium needs a complete set of tools to make energy, vitamins, and building blocks from its environment. Inside Ruegeria MC10, scientists found that many of the genes for these essential metabolic tools are damaged, becoming what we call pseudogenes. This means the tools are less efficient, faulty, or sometimes missing entirely. This makes life in the open ocean very hard. But inside a coral, it's a different story. The coral's tissue is a rich environment, full of ready-made nutrients. Ruegeria MC10's incomplete toolkit mean it naturally prefers to stay where the food is inside the coral. It's evolving to become a permanent, dependent partner. We can still grow it in our labs, but in the wild, its best shot at survival is to stick with its host. The critical question is, does this partner actually help? First, we tested it in the lab. We exposed a model anemone, which works like a test tube coral, to extreme heat. The result? Anemones with Ruegeria MC10 could withstand temperatures 1 full degree Celsius higher than those without. In a warming ocean, that 1 degree is a lifesaver. But the real test was the ocean. We gave our corals their Ruegeria MC10 shield in the nursery and then transplanted them onto the reef. For months, we waited. Then, at the 6-month mark, a severe marine heat wave struck. A massive bleaching event began. The ocean was turning into a bath. The results were stunning. While unprotected corals bleached severely, the corals fortified with Ruegeria MC10 showed dramatically less bleaching. Their color was stronger. Their internal machinery, measured by their photosynthetic efficiency, was still running. Ruegeria MC10 had persisted inside them throughout the 8-month monitoring period, without any reapplication, and created a shield just when it was needed most. The crisis is real, but so is the hope.